the sort of users who've joined previous sessions of the sort of single cell exploration club, and new users as well that are joining these sessions. So for today, I'll be sort of the speaker. My name is Alexis. I'm part of the customer success team at Bioterine, who also sort of support users on sort of utilizing the Bioterine system for very specific case studies in their research pipeline. So the case study today will mainly be focusing on a new analysis, so a new functionality, which is the cancer cell prediction that is found in the bBrowser X Pro system. So the new updated version of bBrowser X. And mainly this particular function, which we'll cover today, will be applied in the context of multiple myeloma. Particularly focusing on how can we identify and characterize very specific malignant hematopoietic stem and also progenitor cells within sort of publicly available data sets that have already been analyzed, but haven't really been identifying these malignant progenitor cells in their studies. So we'll try to sort of use this particular sort of inverse CNV methodology within the system to be able to profile the different transcriptional changes within these malignant sort of progenitor cells in the context, again, of multiple myeloma. Okay. So just as a recap from the last week's session, so we do hold these single cell exploration clubs every week. And last week, my colleagues particularly focused on our new Talk to Data version 5 database. So we have released as of last week this new sort of version 5, which has or is encompassing several more data sets than in the previous version. So over 3,600 data sets just in human. Sort of the human species within the Bioterine system. And we also covered mainly how our team sort of processes these publicly available data sets from different systems, how they're curated in the systems, how we build our own standardized ontology systems to be able to really cross-communicate across different platforms. So for example, utilizing the version 5 human database to apply cell type meta-reference annotations on your own internal data. From there, we also have deconvolution of pseudobulk within the SmartBulk app. So being able to apply sort of these annotations using all of the single cell RNA-seq data and applying it to bulk RNA-seq analysis. And this sort of pipeline also sort of interconnects or sort of integrates within the spatial X or the spatial transcriptomics omics space. So how we can deconvolute annotations within single cell RNA-seq and how can this be applied to be able to understand cell-to-cell interactions within sort of the tissue space. So this was the focus of last week. If you are interested, we do have within our sort of YouTube page all of the recordings of previous sessions. And I just wanted to recap some of the analysis we performed. So for today's sort of focus, the agenda will be sort of broken down into 4 different sections. The first part, which is more to give you an idea what data set we'll be using from the public database, specifically what analysis was already performed from that study and how we're building from that study and applying the cancer sort of prediction model within multiple myeloma. So that will be the first part, understanding that context. The second part will be more how can we apply this new methodology, so this inverse CNV type of pipeline within the bBrowser X Pro system to be able to identify these sort of cancer cells across studies that might have not identified from gene expression alone. The third sort of part of the session will be from that sort of analysis and that prediction result from that study. Are we able to really quantify these cells across different treatments within multiple myeloma? And also, how can we see the proportions from the original cell type annotation, so the healthy progenitor or stem cell-like cells within the study, and how they sort of were annotated as cancer cells, and what is the proportions within this analysis? And then from there, it's more having to do with our downstream features, so differential gene expression within these very specific stem-like progenitor population cells that were characterized as malignant based on this sort of inverse CNV model. Okay. So some context again, before we dive into the actual live demo and how you can perform this analysis in bBrowser X Pro, I did want to give some context on the actual sort of disease model that we'll be focusing on. So particularly this particular sort of hematological malignancy, which is multiple myeloma, is sort of characterized as a plasma cell malignancy, which originates from sort of the bone marrow, and it represents sort of the second most common hematologic cancer across sort of the oncology space. In terms of why we want to focus on this particular sort of hematologic cancer, is because there is normally seen a lot of uncontrolled proliferation and malignant plasma cells that leads to this sort of excessive production of abnormal antibodies. This causes further complication in the patients, and also there's a lot of differences in terms of clones that can be identified within the micro sort of oncology space when it comes to relapse within multiple myeloma patients. And particularly what we see is that even though there are a lot of treatments, particularly looking at stem cell transplantation, we still see that there remains a major sort of clinical bottleneck within these sort of patients and these sort of previous studies. And mainly this has to do with sort of applying traditional sort of regimens, which haven't been really successful in all patients. So about 50% of these patients normally do not respond to these therapies. So there's a fail response, especially in patients that have other exposures to other agents. So the reason why I guess we're focusing is really to understand what are these subclonal sort of specific microenvironmental impacts within multiple myeloma. Can we use sort of these different models to identify these cancer cell populations that might have been previously sort of annotated as sort of healthy progenitor cells within different tissues, or also blood samples as well? So a bit having to do on the specific functionalities that we'll be covering. I also wanted to give you some background on what sort of analysis has been done within the space. So particularly if we look at the immune microenvironment and what is sort of infiltration or genomic alterations in multiple myeloma, what we see is that even though multiple myeloma is primarily a bone marrow sort of malignancy, there are malignant cells that have been, from recent study, have been increasingly detected in peripheral blood as well. So not only sort of tumor tissue. And specifically, this is relevant within very specific conditions of multiple myeloma. So not all sort of subtypes or all sort of disease severities within this context, but very specific sort of clinical conditions. And what is known as these sort of circulating tumor cells within peripheral blood, they often harbor very large scale sort of chromosomal abnormalities, which is something that we'll try to touch base and how we can use these models to identify these sort of chromosomal abnormalities that are not just evident based on gene expression. So normally the user, when they try to analyze these different sort of samples, these different sort of cells within the microenvironment in multiple myeloma, they tend to sort of identify immune cells more broadly than they would cancer cells specifically in blood. So that normally requires more than just sort of the normal clustering analysis or gene expression enrichment. It requires being able to look at the genomic alterations within these chromosomal sort of aberrations in these different samples. So again, given that these multiple myeloma originates in early progenitor cells, it is plausible that these stem-like cells might be sort of changing within mobilized grafts, so very particular treatment scenarios, and they might have very specific oncogenic sort of CNVs that might be retained when they're sort of migrating through these different mobilized graph sort of models. So this is the context that we'll try to really analyze, especially because the study that we'll be using didn't particularly focus on identifying these genomic alterations that might have an effect on sort of relapse in different treatment models, and also looking more broadly on sort of cancer cells that can be identified within sort of circulating or within the blood sort of space. So this is the study that we'll be sort of using for this case study, which is a new data set that has been sort of recently curated in the Talk to Data version 5 database. And just so that all the members or all the users that are joining the session know how to find the study, or they haven't used Talk to Data before, I'll briefly show you how to do that. So when you're logging into the BioTerrain system, you'll see that there are different platforms for very specific pipelines and research needs or questions that you want to answer on your data. So for this case, we'll be mainly sort of mining the public database to see what data sets can we identify that are particularly looking at multiple myeloma or plasma cell myeloma sort of conditions. So when we access the Talk to Data database, this is when you'll see sort of this new version 5, which has been recently released. There are different ways on how to search through the database. One, which is if you already have very particular targets that you want to see where these sort of specific genes are expressed, and you want to validate that across different studies within the system based on your own filters, you can do that as well. But for this scenario, we'll be sort of looking at just the studies within the database to really identify what number of studies can we sort of use or reanalyze to sort of answer other questions. So for this case, what I'm sort of focusing on is particularly multiple myeloma. So normally the ontology system within the Talk to Data, it has been standardized. So that means that it's easier for you to be able to identify very specific samples across different studies based on this sort of trained standardized ontology system. So since multiple myeloma sort of is sort of under a sort of larger umbrella, which is plasma cell malignancies. That's how we would normally be able to identify those studies. So this is where you can sort of select that specific filter. You can add also other filters, which I'll explain as well what those will sort of change. So when we apply the search, this is where you'll see how many studies we have within the system across 2,000 data sets, and 36 studies are particularly sort of focusing on multiple myeloma or other plasma cell malignancies. So this is where you can explore those studies further if you have very specific questions that you want to answer or you want to validate your own target genes that you've seen within your own data set. That's also another sort of question you can answer. And you can go even further from there. So you'll see here that there are different immune cells that these authors have identified already within each of these data sets. And let's say you want to focus on particularly the question on how can we identify these neoplastic cells or these different malignant cells that might have already been or previously annotated as sort of just healthy progenitor cells. So this is another sort of filter that you can add to that granularity. So if we want to look at or focus on particularly sort of these neoplastic cells within these different data sets only, and we want to do a cross-study analysis of these cells across these different sort of models, this is where you can add that sort of cell type filter that we want to focus on. And this will give you exactly what studies have that information that you can use to answer other questions from your own analysis. So this is where you can find the studies, and some of these studies here, one of them will be the one that, again, we'll be using for this particular example. So we'll be, again, using this particular one that looks at sort of mobilization of different treatments, particularly hematopoietic stem cells transplantation within multiple myeloma, and looking at different treatments that can mobilize sort of this transplantation model. Okay. So again, this will now focus on sort of the live demo, how we can perform this analysis and really go into deeper sort of questions on how we can identify these hidden cancer clones in multiple myeloma, which can be mobilized by different grafts, treatment arms using sort of this inverse CNV model or cancer prediction model within the system. Okay. So this is where I'll go into our sort of prediction model and the specific data set in the system. So you can see here that we'll focus on particularly throughout the session, mainly how we can identify transcriptional changes, usually using differential expression model, statistical analysis, and also how we can really predict the cancer cell enrichment or the abundance of these cancer cells within the study. So the feature that we'll be sort of using is this cancer cell prediction that is in beta. So that means that we're still adding a lot of features to this functionality. And really, this is going to be based on your feedback as well after the session. If there's other questions that really weren't answered and you might sort of benefit from these other functionalities within this prediction model that you want to see within the system, you can provide that feedback at the end of the session. So once we go into this prediction model, and I'll zoom in just so you're able to see a bit clear. So in order to be able to perform the analysis, I'll explain a bit what the cancer prediction model is and how you can actually use it, or what processes the pipeline actually runs when you're sort of using this function. So the cancer detection model, what it does is that it's sort of a pipeline within bBrowser X Pro, which tries to sort of provide or allow you to identify these malignant cells in single cell RNA sequencing data. This process, what it does, it tries to sort of begin from the single cell data set. So it groups very specific similar cells using sort of a group-based model on their gene expression profiles. And what this does, it forms these pseudocells within the model. So these pseudocells later on within sort of the model, they help reduce the noise that increases the signal for detecting copy number variations. So what we're trying to really identify is, can we see these copy number variations, but can we also see this reduction in noise within the data, which is quite common in single cell RNA-seq, where you have a lot of noise and you're not sure sort of if these are cancer cells, or is this mainly because of the abundance of certain markers that were used in a panel within the study, and which is sort of not giving a really clear information if these are actually malignant cells. So that's sort of the key analysis. So bBrowser Pro applies sort of this infer sort of CNV model, which is again this sort of cancer detection pipeline. And this is a computational tool that really compares gene expression across genomic regions. So not only how that specific cell is expressing that gene, but how that's compared across different regions in the genome. And this allows to really map the cells in the CNV space and add very specific scores to these cells. Whether they have this sort of threshold that might give sort of indication that these are actually malignant cells that are transforming from these healthy progenitor cells within this context. So if you're interested in learning more about what method do we do use a sort of scanning method, which is the Otsu sort of threshold method, which is more adapted from imaging sort of processing. So it tries to adapt this CNV score segmentation. And this is how you would be able to visualize it. So it tries to really see how the CNV score is sort of within that space without or with sort of pseudocells, and how can these be enriched based on sort of these different sort of gene expression across the genomic region. So that's how we're able to really use this model to really go into what are the genomic alterations that we see within the cells, and how can these be sort of useful in identifying these specific heterogeneity within these cancer subclones. Okay. So from here, I'll show you how you can actually apply the model. So if you want to create a new analysis, there is some sort of information here on what exactly the model is going to do, how the scores will be sort of applied to the different cells. And one thing that you'll need to actually select is that you'll need sort of a specific cell type that would normally be sort of a normal control cell type that will be used to really compare across these different sort of genomic alterations to really identify which ones have this sort of CNB instability. And can we see very specific subclones within very specific cells compared to these sort of normal healthy cells that you would normally see in the study. So this is where it will require you some, I guess, background in the type of tissue that you're working with or the type of disease that you're working with, and comparing it to a very specific sort of control cell type. So in this case, within the study, there was an explanation that this specific patient-specific cluster was more related to this sort of healthy related remodeling. So this is sort of the cluster that I used as a reference to be able to identify if these other progenitor cells are actually changing based on sort of the grafts, so the treatments that are being used within these multiple myeloma patients that are really driving this sort of I guess, the stem-like cell malignancy within these progenitor cells. So this is sort of where you'll need to select the cell. You can add other advanced parameters based on sort of the UMAP or the analysis that you're performing. Once you add those parameters and you add your control group that will be compared and to be able to identify that enrichment, this is where you can run the analysis from there. So I have a specific sort of previous result just because it might take a few minutes to load. So when we see sort of this result, what we can see is that previously the data set did not really look at very specific cancer cells. So their main focus was really looking at how can they mobilize CD34 hematopoietic stem and progenitor cells across different sort of treatment strategies, which is the moxifluoride or the sort of granulocyte CSF treatment within these patients. And they were really trying to really immunophenotype very specific immune cell populations across these different treatments, but not particularly looking at how these progenitor subtypes might be actually sort of transitioning to a malignant transformation that might be due to their sort of genomic level or the CNV, again, genomic alterations that might be within the genome. So that's where we're adding this sort of added analysis. So we can see here that the prediction model was able to identify clearly and a very specific cluster of these cancer cells. Normally, the actual default or the cutoff range is automatically selected based on sort of the higher values and which ones are actually considered cancer cells. From here, there's other downstream analysis that you can actually gather or other results that you can really sort of understand from this analysis. So not only the ability to see, okay, where is the cluster of these cancer cells based on these sort of genomic alterations, but what type of genomic alterations can we actually visualize? So when you go into the heat map functionality, which is this one right here, if I go into the slide, this is where you're able to actually see the CNV space of these pseudocells and what is sort of these chromosomal aberrations that you're seeing within these specific contexts or these samples within the study. So again, if we go into the heat map, this is where you'll see that particularly within this cancer cluster that was sort of identified as cancer cells that were originally healthy progenitor cells annotated in the study, we can see that these specific cells actually have very specific chromosomal aberrations compared to the normal progenitor cells in these samples within these multiple myeloma patients. So if we want to sort of gather more information here, we will be sort of adding more functionalities as sort of the months go by in terms of what type of chromosomal aberrations do we see. So based on other studies, some of the ones that are quite common in multiple myeloma are sort of the gain of the 1q or specific deletions of certain chromosomes that might be sort of linking to this sort of driver event or these differences within these cancer cells that are transitioning. So this is something that we'll be adding to this feature, so you're able to go deeper into what sort of chromosomal aberrations can we see in these studies. Okay. From here, the next sort of function that you can also visualize within the system is sort of this lineage tree. So this is more having to do with how can we see the different sort of ancestral tree from these cell types, and can we see an evolution of these cancer cells within very specific regions or clusters. So this is another analysis that you can also sort of run when you're running the cancer cell prediction, and you can automatically create this sort of evolution tree. And what can we see here is that the lower right branches show mainly these densely packed dark red knots. So what that means is that these represent a very dominant genomic complex clone, possibly responsible for the disease progression or this sort of relapse within sort of these treatment groups. And what we can see upstream is that we see these lighter clones, which really reflects this early stage or this ancestral clones, which might be more resembling the normal sort of progenitor cells that were annotated within the study. So you can clearly see that there is a very clear difference where you have these sort of normal progenitor cells that are transitioning into these actual malignant cells that have these sort of clonal instabilities within the samples. So these are analysis again within just the functionality. And one thing I would recommend if you have these sort of UMAP and this annotation, in order for you to run other downstream analysis where you're actually using these annotated cells from the prediction model, make sure that you actually save the prediction result as sort of its own metadata. That way you're able to sort of look at the actual distribution of the scores if that's something that you want to see how these specific scores are distributed across different clusters, or if you actually want to use the label to run differential gene expression within these cells and actually see if there is any treatment differences within sort of these mobilization graphs. So once you save the metadata, you'll be able to go back within sort of the original UMAP, and you'll be able to see that within the private metadata, so any metadata that you're generating on top of the original study, you'll be able to see those annotations. So in this case, the cancer cell prediction CNV scores across sort of the UMAP, and also the actual label so that you're able to subcluster specifically those cancer cells and compare them across different treatment sort of graph groups. So from here, one thing I wanted to sort of mention is how can we actually visualize the proportions within these different cells? So now that we were able to run the prediction result, how can we quantify the proportions of these cancer cells across the originally sort of annotated cell types to see where the abundance is? So what sort of healthy progenitor cells have been transitioning to this other sort of cancer CNV instability sort of model? And can we see any differences across these cells? So in order to do that, once you have that label, you can go to this visualization functionality, which allows you to create these different cell proportion charts or looking at the composition, so the number of cells and the sort of percentage of cells within different clusters. So this is quite a useful tool, especially if you already have the prediction. So let's say we want to focus on the multiple myeloma samples, and we want to look at the actual cancer cell prediction. So what cancer cells are relevant within the previously annotated cell types. And I'll bring up the author's original cell subtypes to see where is sort of the higher proportion, or what is the higher percentage of these sort of healthy progenitor cells that are now relabeled as cancer. So what we can see here is that particularly if we just focus on the multiple myeloma group and we're focusing on particularly the cancer group, what we can see, and if I zoom in a bit clearer here, is that we see an abundance specifically in the hematopoietic stem cells and also the common lymphoid progenitor cells. So this is where we see the higher sort of percentage. So we can see that there's 4.9% cancer cells that are sort of distributed within the sort of previously annotated healthy sort of progenitor cell lineages. And these are other contexts that you can get from the data. So to actually see, okay, what annotation differences can we see and what specific cell types are actually transitioning within these different alterations in the sample. So if we go into the next sort of question is how can these cancer cells differentiate across different treatments? So there is a lot of treatments that are being used within the study, particularly different graph treatment groups. So what we want to see is that is there any distribution differences or any quantification differences across these different treatments, but particularly focusing on our cancer cells that were predicted within the model. So what we can see here again, if we use the composition chart and we add sort of the multiple myeloma samples, we see our prediction. So the normal progenitor stem-like cells and the actual sort of predicted cancer cells based on the CNV structural abnormalities, we can see that there is a clear difference in terms of the treatments. So what can we see in terms of the treatments is that compared to the treatments that are actually sort of a combination of different treatments, so using sort of the moxinofortide with the GCSF or a different sort of agent, we can see that there's actually a higher abundance percentage of cells within the cancer group that are mainly coming from just the granulocyte CSF treatment group. So this means that there is sort of differences in terms of sort of the, I guess, combined treatment as just the sort of one line treatment group within these cancer cells that are leading to this sort of clonal instability and this sort of structural abnormalities in these cells. Okay, so the next step really is how can we gather more information from this? So we have our identified cancer cells from the INFR-CNV model. We were able to see sort of comparisons of these proportional changes within previously annotated cell types within the study, and also looking at if there's any proportional differences across different treatments within the study, particularly looking at these predicted cancer cells. So the next step is, can we characterize or can we look at very specific transcriptional characterizations within these cells, particularly these malignant sort of hematopoietic stem cells that have a higher proportion within the GCSF treatment. So we want to focus on, can we gather very specific targets that might be leading to these structural changes, and also sort of showing this sort of response difference as well to a very specific treatment as compared to a combined treatment group. So the groups that we'll be sort of using for the differential expression model will be looking at the CNV predicted cancerous hematopoietic stem cells within specifically this treatment group, and comparing that to the normal progenitor cells within sort of this treatment group to see if there's any transcriptional changes that we can gather from the differential gene expression. So in order to do that, once we have sort of our clusters, or once we have these cells that we want to focus on, the next step is to perform differential gene expression. So an easier way to actually know what groups you're going to use and what specific cells you're going to use is to actually map out the groups that you want to sort of focus on. So the first thing is, again, we're focusing on samples only in multiple myeloma. I want to focus on my actual cancer predicted cells, so the actual hematopoietic stem cells that were sort of labeled as having this sort of structural abnormality, and I want to see this treatment overlay as well. So once we add these labels or these metadata sort of groups, this is where you can perform differential gene expression, where you're focusing on a very specific treatment arm within the cancer predicted cells to look at other transcriptional changes. So when you go into differential gene expression, this is where you'll see that it automatically allows you to select what your group 1 is. So my group 1 will be focusing on the predicted cancer cells, but specifically these cancer cells that have a higher proportion within the granulocyte CSF treatment. So that will be my first group. The second group will be how can we see very specific transcriptional changes in the normal progenitor cells, particularly again in the same treatment group. So this is where you can select the groups, and you can actually, from there, you can run the submission, which will allow you to select what statistical model you will use to run the differential gene expression analysis, and also what title or what labels you're going to add to those cells as well. Okay. So in order to show you some of the analysis that I ran previously, because it will take some time to actually perform these different figures, is that we can automatically sort of identify very specific upregulated genes within these different sort of cancer group models. So these different hematopoietic stem cells in the cancerous transitional state compared to the healthy progenitor cell state. So we can go to the upregulation to see what sort of markers are sort of driving these changes. And we can also select the top 20 genes to actually see what are the top 20 genes that we can gather within these different groups. So if we go to heat map, this is where you can actually look at the very specific targets that are driving these sort of hematopoietic stem cells to have this sort of cancerous transitional state, and also these sort of CNV aberrations as well. So if I go into the slides, which will make it easier to explain, some of the genes that we can gather that have been shown to have sort of an effect is this NEAT1. We can also see that PTEN has been upregulated within these sort of cancer hematopoietic stem cells. We can also see other genes like CD83. And this is, again, analysis that you can perform to gather more results from there. So some of these genes are quite important. So what we can see is that these malignant cells, particularly within this treatment group, so the granulocyte CSF sort of transplantation graph model, we can see that the NEAT1, which is an immune evasion sort of gene, is upregulated. We can also see that the NFAT5, which is a stress survival gene, is also upregulated within these cells, and also PTN, which is more sort of contributing to this residual disease and relapse within this transplant model. So there's a lot of analysis and a lot of insights that you can gather once we run the infer model, and you can use these sort of prediction results to run other downstream analysis from there. Specifically, if you have different treatment models that you want to really analyze further. Okay, so I guess this concludes a bit of the analysis that we can perform in the system, particularly looking at the cancer prediction model, so the infer-CNV, which allows you to find these structural abnormalities within the genome. How you can use those cells to really look at proportional changes. And from there, how can we train these to actually look at transcriptional sort of abnormalities by looking at upregulation of certain genes within these different groups that were selected. So some of the results that we can gather is that the infer-CNV was identified in 5% of these mobilized cells as cancerous, which wasn't particularly sort of identified in the original sort of clinical trial. And this resembles within mainly the hematopoietic stem cells. So across other cell types or other immune cells, the hematopoietic stem cells are the ones that are sort of having this transitional state and these particular structural abnormalities that are aberrant, and they're sort of different from these normal-like clones that you would see in other sort of progenitor cells within these different graph models. The second sort of result that we were able to see by using different visualization tools in bBrowser X Pro is that the GCSF particular treatment alone was mobilized and has the highest proportion within these cancer-like cells, while the other sort of combined treatment mobilized graph models have more total cells but fewer malignant ones or fewer malignant clones. So this suggests a greater sort of selectivity and also potentially a safer graph composition within the model. The third result, once we performed differential gene expression within those cancer-like cells, we were able to see that within this specific treatment group, there is a very clear upregulation of certain genes that are linked to immune evasion, stress survival, that are very important when it comes to sort of persistence and also relapse in certain treatments, or that can really explain why 50% of those patients are not really responding to that treatment. So that's again some of the results that we were able to sort of cover today. My colleague is sort of following up with a survey of this session. So we want to gather some insights if this session was useful for you to be able to now perform your own analysis on the system and be able to use this new cancer prediction model within bBrowser X Pro. So please, if you can fill out the sort of survey to get some of your feedback and also to prepare other sessions later on that might be covering other models that might be that you might be focusing on. Okay. So I'll just leave a few seconds so you can actually fill out the survey. If you have any questions, I can also sort of answer those questions as well, or you can send them to our direct email so we can follow up with you as well on a sort of separate session. Okay. Okay, so I don't see any Q&A, but I'll add in the chat the actual email that you can follow up in case you're a new user and you haven't actually been using Biotrain recently and you need a recap of all the functionalities or how to upload your data. Just let us know so you can reach out to that email and you can schedule a one-to-one session so we can go through these features, but particularly focusing on your specific case study and very specific research questions that you want to answer. Okay. Well, thank you everyone for joining. So I think we'll close the session for now. If you have any other questions, again, reach out to us. We're happy to sort of support you. And thank you again for joining the session and sort of being very attentive to these different sort of single cell exploration club case studies. Okay, so thank you everyone.